No, we don't. Hold on, because there's no thymine. Disregard. So we have U, A, C. Then we have C, G, C. Catch my drift? So you will do that for the anti-codon. It's the same rules. There's no, don't be Mrs. Paris and put a thymine in there. It's really easy to do that. So make sure you check yourself. So you will keep on. So far, are we okay? I'm coming back over here to check. So far, are we good? Okay. Remember, just do your codons, make sure your uracils are replaced instead of the thymines. See, I made that mistake like right off the bat and I caught myself because I want to just do I want to do DNA replication. That's where my brain is. So you just have to work slowly. And because it's an easy thing to do. It's easy, but it's easy to make a mistake, if that makes sense, right? You can get caught up real easily. So now here's where we get tripped up. And it's not even really a trip up. When we do this part, meaning the codon chart, which is the thing here at the bottom, we don't use the transfer RNA. We use this sequence. I'm going to get my highlighter. We use the mRNA to do this. The transfer RNA is the anticodon and is which is what helps it be transported outside of the cell to be matched to its complementary base. The messenger RNA is what literally translates it to the amino acid. This is the translation piece. This is the transcribing, where it basically transports it to a different location. That's what you need to know. So when you do this, or when you do the amino acid, when you're making the singular beads, so to speak, you're using the messenger RNA. And what you do is you look at, here's the first codon, or the first, excuse me, nucleus. Well, this is the first codon, but the first base will be highlighted to this first base. So this is your first base here. Your second base, we'll use yellow, is these letters, your second base. And then your third base is the third letter of the codon, and that is over here. Okay? So when we're trying to figure out, now, I also just want to put a disclaimer that these amino acids are all abbreviated by the first three letters, like let's say MET, that is short for methionine, right? Nobody wants to write out every single amino acid, so that's why they're abbreviated to the first three letters, just an FYI. So when we do this, here's what we do. We look at the A, so we look for A, so this is our first base. So here's A, right? And then we go to the second base, which is U, so we go to the second one, which is here, but we're down here, right? So we're A, and then we have U, and then the third letter is G. So it is met. So the first, and if you look on your Florida virtual, methionine is actually a start codon. There are different codons. There are start and stop codons because that tells the DNA molecule when to start and stop translation and transcription, okay? So let's do another one. We have G, then we have C, so we're gonna go over one more, and then we have G. So we have alanine. Let's do a third one. We have AGG. So we have our first base A, then we have G, which is over here, and then G. We have arginine, which is ARG. Can I have somebody do the next one for me? Can I have somebody do CGG? Yep. Can I have somebody do GAG? Yep. Can I have somebody do the next one? Yep. What about the next one? What about the second to the last one? Yep. And then what about the last one? Yep, it is stop. How are we feeling about that? Are you talking about the Florida virtual notes?
Yeah.
I completely ditched Florida virtual for this unit. So that chart is not the one that I'm using. I kind of went rogue because I hate that. I don't really like the unit. Some of the stuff is good. The last slide of it, like kind of condensing everything, slide 8, is good. That's a good slide where it just condenses everything for you. The codon chart that I want you to use will be the one that you see here. I'll post one on Canvas because that one on Florida Virtual is annoying anyway to use, I think. I think this one's easier. How are you guys feeling about this? So now, for homework, you have, I did number 2 with you. That's what we did. Now you have the other ones, and then you have some questions. Well, mine's on the back because I printed mine out today because let me tell you, Mrs. Harris's eyes are horrific and I need it to be on a piece of paper. So now I was a little tricky and rude to you. And like number 1, I gave you the mRNA sequence. So then you need to do, so if you can, now you have to work kind of backwards, right? And then at one point, I just give you the amino acid sequence. So this is homework for tonight and tomorrow. I think that gives you plenty to do and to digest over the next, day or so. And the questions on the back, 6, 7, 8, 9, and 10, or I should say on the second page, I haven't gone over stuff formally. Like I've gone over number 6, like what are the 3 differences between RNA and DNA? But all of these you can find in the Florida Virtual notes. I'm still going off of Florida Virtual, but there's a couple things that I'm not, like the codon chart. I don't like it. And the reason it's not, I didn't use it. So I will post, actually, hold that thought for a second. If you want, before I set you free, shared drives. No, I'm going to do that for class on Thursday, Sophia. I'm going to use that note sheet. The questions I might have you do on your own, but I'm going to do question 1 with you. That's, or I'll assign it on Thursday. It's not due. I haven't decided yet, but I want you to concentrate on the sheet that we just went over. Now, here are 2 versions. These are in the shared drive, ladies and gentlemen. I'm just giving you direct, they're in the shared notebook drive. I'm just giving you direct links to them. So if you want to click on them and then save them to your drive or whatever you want to do, there's 2 different codon charts. One, ladies and gentlemen, is it spells out the complete amino acid, and one is abbreviated. Are they working for you? Can you open them? Yes, no. Okay, well you can find them in there. They're in there. I had to, mine are like really embedded, but you'll see the codon chart. Those are the ones that I've been using. So just to be clear, this sheet here that I shared with you, right? That Sophia was talking about, I will be going over this part. This is how transcription and translation is done. Like, that's the biology of it. So I will be going over that on Thursday with you. So you can work on this if you want. Some of the questions you can answer if you'd like. It's in your notes, but I will be going over this formally. This is homework, and I'd like to, on Thursday, go over some of this with you to make sure that you have a full understanding of transcription and translation, like physically doing it and transcribing a DNA sequence, molecule sequence, and then I'll do the biology behind it. And then Friday, we'll do mutations, right? We can't just learn about things going 100% right. We have to add a curveball in when bad things happen in those sequences. Like when we have an additional